Hello, and welcome to Claret Bio's fourth installment of the Serious Talk. At Claret, we specialize in the development of NGS library preparation and analysis tools that empower researchers to get the best data from their most challenging sample. Our flagship product, Seriously, is a single-reaction, single-stranded DNA library prep kit known for producing complex libraries. It has found applications in a diverse research areas ranging from ancient DNA and FFPE to cell-free DNA and oligonucleotide samples. Seriously works in a one-step combined phosphorylation ligation step that prepares template DNA molecules for ligation without the need for end polishing. Additionally, we are thrilled to announce some exciting news about our upcoming product launch this fall, the Really Are None RNA-seq Library Prep Kit. Designed to streamline laboratory processes, Really Are None generates truly directional libraries without the need for second-strand synthesis. Additionally, it incorporates a downstream ribodepletion method, ensuring that researchers can obtain high-quality data from even the most challenging samples. If you'd like to learn more information on this upcoming launch, please don't hesitate to reach out to us after this webinar or contact us at info@claretbio.com. Now, without further ado, I'm thrilled to introduce our guest speaker, Dr. Lydia Freddolino, who specializes in the molecular mechanisms driving bacterial behavior. Dr. Lydia Freddolino is currently an associate professor in the Department of Biological Chemistry and the Department of Computational Medicine and Bioinformatics at the University of Michigan Medical School. Her lab develops and applies high-throughput sequencing methods to study the interplay between bacterial chromosomal structure and gene regulation, with an emphasis on the roles of highly abundant histone-like proteins called nucleoid-associated proteins in shaping bacterial stress responses and regulation of virulence genes. We're glad to have you here with us today, Dr. Freddolino, and we look forward to your presentation on genome-wide profiling of RNA-DNA hybrids and bacteria, revealing the central role of R loops in shaping bacterial genome evolution.
Thank you very much for the introduction, and it's great to be here. So as she said, I'm going to talk about some work we've been doing on looking at the effects of RNA-DNA hybrids on chromosomal structure in bacteria. Before I get into any of the science, though, just a couple of disclosures and disclaimers. As far as any relevant affiliations, I'm an editorial board member of Scientific Reports and EcoCell Plus. I have no affiliation with Claret, and I'm not getting any compensation from them. And I want to be very clear, I'm going to talk about what we use in our research. This should not be taken as an endorsement of any particular product. So my lab in general is interested in a bunch of different topic areas, and they fall into two major categories. One is bacterial chromatin, as I call it, and gene regulation. And I don't use the phrase bacterial chromatin lightly. I know it's not one that people think a lot about, but I think I am going to be able to show today why this is relevant. And then on the other side, we also do a lot of work on protein structure and function prediction. Let's see, it's video on now. Let's try that. Still says I cannot turn my video on. I'll try again in a moment. So what I'm going to do for today, at least, is I'm going to talk a bit about this first part, the angle of bacterial chromatin and gene regulation, and then zoom into our work on DNA-RNA hybrids, where the ability to do single-stranded DNA sequencing preparation has actually really played an essential role in our biological findings. So before I actually go into the data, I'd also like to just thank the people who were involved in this. So two folks in my lab, Rebecca Hurdo and Jeremy Schrader, did a lot of the work, Rebecca on the experimental side and Jeremy on the computational side. And we also had, this is an ongoing collaboration with Professor Eliel Simmons, who also runs a lab here at the University of Michigan. So when I think about bacterial chromosomal structure, and this will get us to the RNA-DNA hybrids, you'll just have to be a bit patient with me. One of the key approaches that we have used to probe chromatin structure in bacteria is an approach called IPOD-HR. It stands for in vivo protein occupancy display at high resolution. And this emerged from a problem that I had noted, which is bacteria aren't really amenable to ATAC-seq or related methods, but if we're interested in tracking a complex response, or some kind of regulatory response in bacteria, there are hundreds of transcription factors we would need to track. And so it is not practical to do the number of ChIP-seq experiments that you need to do to track them all. So what we wanted instead was a method that lets us see where is their protein bound on the chromosome and use that to figure out what's going on with their regulatory logic under different conditions. So the way this method works, it begins looking like a ChIP-seq experiment. You grow cells under some condition of interest, you blow the cells up, shred the DNA down to small footprints, and instead of at this point doing an immunoprecipitation with an antibody as you would do for ChIP, you instead throw this into a phenylchloroform extraction, and under the correct buffer conditions, you get the formation of an interface layer that is enriched for the protein-DNA complexes. You can recover the DNA from that layer and sequence it and compare it to an input sample and get a profile for where there was protein bound on the chromosome. So to give an idea of what this looks like when we already know the right answer, this is one of my favorite regulatory regions in E. coli. It's upstream of a gene called PRC, and we know what should be happening here. We know that there is a binding site for the transcriptional regulator PRR present right here. And when there is rich medium, purines present in the media, then PRR should be bound and repress transcription here. If you take away the purines, then the PRR should go away and allow transcription to occur. And that's precisely what we see. So when you grow the cells in rich media, we get a nice peak in occupancy, and this is our nonspecific occupancy signal that matches up really well with the known PRR binding site. That peak goes away if we grow the cells in minimal media when the PRR should not bind. It also goes away if we delete PRR, but grow the cells in rich medium. And so that's great. We were able to recover what we already knew about the regulation at this particular promoter. But what's especially helpful is we didn't have to decide ahead of time that we were looking at PRR or at this particular location. We're getting the same information on DNA-protein interactions genome-wide. And there are a lot of interesting things we've been able to do with this data, but I think the one I want to focus on for right now comes from zooming out a bit and taking a global perspective at the occupancy profile. And so if we zoom out and just look at this over longer length scales, we get the signal that's shown in blue. This is looking at a roughly 50 kilobase region of the E. coli chromosome, and we're smoothing the occupancy trace out a bit. So the blue that we have here is our iPod protein occupancy. So this is total protein. And then the orange is RNA-seq that's been gathered under the same condition. And two really striking features emerge here pretty quickly. First, we have some regions like this that we call extended protein occupancy domains, which are large regions of high protein occupancy. Second, if we look at where the extended protein occupancy domains are, they seem fairly anti-correlated with transcription in that the regions that have a lot of high protein occupancy have not very much RNA. The regions that are being highly expressed don't have a lot of protein occupancy. So this is already really exciting to us because if there are these high protein occupancy, transcriptionally silent regions on the genome, it starts to smell a bit like heterochromatin only in bacteria. But this also leads us to what I would call a chicken and egg question, which is, are the highly protein occupied regions just highly protein occupied because there's no transcription? Or are they actually silencing contexts on a bacterial chromosome that would also silence any other genes that ended up in them? So to address that question, we did a high throughput reporter assay. We designed the reporter construct that is shown here. It has an antibiotic resistance marker, which we can flip out. It has a promoter that is inducible, and it is always going to be the same promoter, regardless of which copy of this you're looking at. That's very important. There's a reporter gene, and then there is a unique barcode on every molecule of this. So we construct a reporter library using TN5 transposase, where we randomly integrate different barcoded copies of this construct into the E. coli chromosome. And it gives us a library where every single cell has the same reporter with just a different barcode integrated at a different location. And then we can match the barcodes to locations by using just transposon footprinting. And then we can ask the question, how well can you induce this reporter as a function of where it is located in the genome? So we induce the reporter, we purify RNA and DNA from the induced cells, and then we can just quantify what is the RNA to DNA ratio dependent upon position in the cells. And so what that gives us, and we look at this for 144,000 different uniquely trackable integrants, so that's roughly 1 every 40 base pairs along the E. coli genome. And here's the signal that we get. The y-axis here is what we call the transcriptional propensity. That is how transcribable is our reporter. The x-axis is the position along the genome. And it becomes very quickly apparent that there's a lot of dynamic range here. There are some places like here and here and here, where the reporter is very easily transcribable if it inserted in those locations. There are others, troughs in this profile, like here and here and here, where if the reporter landed, it is basically silenced. And so if we look separately at what is the transcriptional propensity in the EPODs versus elsewhere, we see that the EPODs tend to be transcriptionally silencing relative to the rest of the chromosome. And so that already answers our chicken and egg question. It looks like these are not just statically silent, but silencing regions of the chromosome that will also act on reporters that land in them. And so we've done a bit more work to unravel what are the rules that make a region activating or silencing. And so one of the strongest features that was correlated with regions being a silencing region or an extended protein occupancy domain is the AT content, where silenced regions tend to have higher AT content. And so in order to see whether we could make this causal, we made synthetic flanking regions for our reporter that were either low GC content or high GC content. And then we integrated these artificially flanked reporter regions, and these are 1 kilobase flanks that you have on the reporter. We integrated these either at a high transcriptional propensity or a low transcriptional propensity site. And so what we see is if we look in a region that is intrinsically high propensity, that's the thing I've shown here as no flank, that is the NFI locus. If we put on 1 kilobase flanks that are high AT, low GC, we end up silencing that reporter, even though it's going into what should be a high propensity site. On the other hand, if we have low AT, high GC flanks, then it ends up transcriptionally active still. And on the flip side of things, if we integrate at a normally silenced position, so this is the propensity we get at one of our silent sites. I've changed the y-axis here to make it easier to see now. If we add on high AT flanks, it makes it even more silent. If we add on high GC flanks, it partially de-represses the integration. So this is actually very important because we see, first of all, that people in fact need to be paying attention to the immediate chromosomal context of any ectopic thing they want to express in bacteria, because it's subject to this additional layer of regulation due to the chromatin structure in the area. And second, that we can actually override the local rules. Oh, hey, look at that. My video actually works. Amazing. Thank you very much. So we can actually override the local rules by changing the AT content immediately flanking our reporter. So if we look at the spectrum of things that impact transcriptional propensity in our library experiments, another major factor is played by ribosomal RNA operons. And so what we observed is that anything that is near a ribosomal RNA operon tends to have a high transcriptional propensity. And so given some of the things we know about E. coli chromatin biology, the reasonable hypothesis was that this was because the ribosomal RNAs in E. coli tend to co-localize in the cell and form sort of a protonucleolus, which we thought might be acting as a transcriptional factory. And so then just the recruitment of RNA polymerase around a ribosomal RNA might also make it easier to transcribe other genes that are near that ribosomal RNA. At least that was the theory. So in order to test that, we did a set of experiments where we took our reporter and we integrated it either next to a ribosomal RNA, so I'm going to call that int, or replacing an entire ribosomal RNA, and I'm going to call that KO. And so, oops, wrong direction, sorry. So if we look at the fluorescence that we observed from this reporter, if we just integrate at the RRNG ribosomal RNA, this is one of E. coli's 7 ribosomal RNA operons, we can see a certain level of fluorescence. If we instead knock out the entire ribosomal RNA operon and replace it with our reporter, and we even get rid of the ribosomal RNA promoter, we still see the same level of fluorescence. So this was actually very surprising to us, and it shows that it is not just the ribosomal RNA transcription that is driving the high propensity in this region, but rather that is something about the neighborhood around the ribosomal RNA that's driving the high transcriptional propensity here. And just moving on a bit, we see the same sort of pattern with integrations near ribosomal RNA H. And an even more surprising finding was if we integrate next to a ribosomal RNA and then delete one of the other ribosomal RNA operons, we're actually able to further increase the expression. And this is an effect we're still exploring. So there's still a lot of richness that needs to be figured out about what is controlling this landscape of position-dependent transcription in E. coli. In some cases, however, we do have a pretty good idea of what is going on. And so one example is if we look at some of our EPODs, we know from prior data what DNA binding proteins tend to bind in those regions. And so one example is around a locus called WA. And so I'm showing that over here. Here's our extended protein occupancy domain in the presence of a set of genes that encode for enzymes that modify the cell wall of E. coli. And so we can see just in wild type cells, this is our total protein occupancy profile. And the region that I'm highlighting here is the extended protein occupancy domain. You can see that this is essentially a wall of protein occupancy, and it's also transcriptionally silenced if we look at RNA-seq data. Now we know that there is a protein HNS that tends to bind in this region. We also know HNS has a paralog called STPA that fulfills similar functions in cells. And if we just delete HNS, we see there is a quantitative reduction in the signal here, but we still have silencing. We still have this wall of occupancy, and RNA polymerase still cannot get in. If we just delete STPA, it also doesn't do a whole lot. On the other hand, if we delete both HNS and STPA, then we see a massive loss of protein occupancy in this region and also transcriptional de-repression, and we start to get expression of the genes in this region. And so this is, I think, a really nice example of this kind of bacterial heterochromatin I was talking about earlier, where we have a large region of the genome that is silenced under most conditions. The silencing can be lifted either by deleting the factors that are acting there or by some environmental conditions. However, there are also some locations in the genome where we see these extended protein occupancy domains, and it is not HNS. There must be some other players involved. And so in order to identify some of those regions, we took a look at if we delete individual nucleoid-associated proteins, which are sort of like the histone analog in bacteria, which of them can we find that are strongly correlated with each other, which would suggest that they might be silencing the same regions of the genome. And here's one example we observed where if we delete FIS or we delete HFQ individually, these are two of the nucleoid-associated proteins, then we see highly correlated changes in the ability of RNA polymerase to access different parts of the genome. And so we then tried to make a double mutant of HFQ and FIS, and something surprising happened. What I'm showing here is the efficiency with which we can make a knockout. This is on a log scale. This is just deleting FIS and then deleting a control gene, CSPE. And so this gives us an idea of the efficiency of our transductions in general. And this is deleting FIS and then deleting HFQ. And so it turns out we are not able to make the FIS HFQ double mutant. There's a very low efficiency here. And when we restreak these cells and try to grow them further, they simply die. And so somehow there is a synthetic lethality between FIS and HFQ. And to figure out what is causing that, we took a look at the classes of genes that show up in extended protein occupancy domains. And it turns out that there are enrichments for potentially harmful mobile genetic elements. We see a lot of transposons in these regions. We see a lot of genes associated with cytolysis, which is coming from prophages or integrated viruses in this genome. And this, as an aside, is another way that this bacterial heterochromatin I'm talking about very much resembles eukaryotic heterochromatin, and it seems to be acting as a sort of genome defense. So since we've observed these enriched classes of functions in the extended protein occupancy domains, we could take advantage of a strain of E. coli called MDS42. MDS42 is one of our classic lab strains from which all of the identifiable prophages and mobile genetic elements have been deleted. And so when we take MDS42 and then we try making the double knockout, we are able to make that with high efficiency. So here is now making the FIS HFQ double mutant in the MDS42 background, and it works just fine. And what is probably going on is if we look at our protein occupancy profiles in a couple of regions that are prophages that are deleted in MDS42, we're able to find places where we see effects of both HFQ and FIS deletion individually. So in blue here is our total protein occupancy in wild type cells, red is deleting FIS, green is deleting HFQ. So both proteins appear to play a role here. And so we speculated that regions that look like this, and we identified 3 of them in the genome, are probably being de-repressed in the FIS HFQ double mutant. And sure enough, if we delete either R1 or R3, which are 2 of these regions, and they correspond to lambdoid prophages, we recover the ability to make the FIS HFQ double knockout. And it turns out that R2 is also a lambdoid prophage, but it lacks an intact lysis operon, and so it lacks the ability to kill the cells. But the presence of this FIS and HFQ tandem silencing is necessary to keep the other lambdoid prophages quiet and prevent the cells from dying. And so from a bit more work that I don't have time to talk about today, we've really been able to identify 3 categories of extended protein occupancy domains in E. coli. One class that is HNS and STPA dependent, like some of the ones I was showing before. These tend to regulate virulence genes and host interactions, and I would refer to these as sort of an analog of facultative heterochromatin, because they are de-repressed under some conditions that are arguably resembling developmental switches in bacteria, things like the activation of motility or of surface adherence. There's a separate class that is dependent upon FIS and HFQ, and that was like the ones I was just showing a moment ago. And these I would refer to as an analog of constitutive heterochromatin, in that they tend to silence genes that are otherwise likely to be harmful to the cells under most conditions. And finally, we have a category that's dependent upon 2 factors called IHF and HNS, and these appear to largely regulate metabolic genes. And just for the sake of time, I am going to skip a bit of data here, but I want to show you an interesting facet of that last category of extended protein occupancy domains, the ones that do metabolic regulation, because it shows just how tightly the E. coli regulate their chromatin and how this is used for gene regulation. And so what I'm showing right here is one of the regions of the genome that is silenced by an extended protein occupancy domain. We know this contains a set of genes that are involved in metabolism in metabolism of an obscure carbon source called idenate. And so normally this region is silenced by protein occupancy. If we grow the cells on KDG, which gets metabolized through the idenate pathway, we see a dissipation of this occupancy, and then the cells are able to transcribe this region. And if we follow a full cycle, and then we go, so we went from no KDG to KDG media to no KDG media again. If we look at the pattern of occupancy that we see, the light blue is prior to exposure, the red is during exposure, the dark blue is after exposure. And so they perfectly open and then reclose this region of the chromosome, dependent upon the environmental stress that they are encountering. And we see the same kinds of changes in RNA-seq data, where if we look at the genes in this region, they start at a baseline level, they go up, and then they go right back down to where they started when the region is re-silenced. And there are very few changes genome-wide. This appears to be a localized change in chromosomal structure in this region that is giving rise to the behavior that we are seeing. And so at this point, we've been able to apply the iPod method across a bunch of different bacterial species, and we essentially always see the same story in terms of there are heterochromatin-like regions that are silent in every bacterium we've looked at. And so here, for example, is some data in Bacillus subtilis. These are our protein occupancy profiles. In blue, we see regions of extended protein occupancy. The green is RNA polymerase, and we tend to see that the extended protein occupancy domains are silent. We see the same kinds of patterns in Streptomyces, where a lot of antibiotic biosynthetic clusters are regulated by extended protein occupancy domains. We see this in Borrelia burgdorferi, an important pathogen. We see some nice regions of high protein occupancy that also look heterochromatinized. And this particular region is involved in antigenic switching in Borrelia when it's inside the host. And so if we take a step back and just think about what we've learned from this, we see that looking at protein occupancy genome-wide, there are many different classes of occupancy that show up across the E. coli chromosome. They look like functional analogs, at least of heterochromatin. This shows up in every bacterium we've looked at, and they can respond to many environmental stimuli, although some of them appear to be constitutively silenced. And I didn't have time to talk today about the fact that phase separation also appears to play a role here. Now, I've been talking a lot about the interplay between chromatin structure and transcription, but if we zoom in on transcription, there's a separate type of structure that actually appears to interact in a very interesting way with the protein occupancy landscape. And that happens to be a category of entities referred to as RNA-DNA hybrids. And so these arise from a couple of different contexts in cells. For example, in the process of transcription, there obviously is an RNA-DNA hybrid that has to form. If that hybrid persists for too long after the transcription bubble passes, it turns into what's called an R loop, which can end up being problematic in ways I'll talk about in a moment. There are other important places where RNA-DNA hybrids show up. For example, Okazaki priming during DNA replication. They're involved in Ig class switching in the immune system. But the difficulty that comes up is RNA-DNA hybrids can lead to genomic instability if they are not managed carefully. So here, for example, is a schematic of an R loop that might persist in the wake of a transcription bubble. And the problem that arises is because there is a single-stranded region of DNA left in this structure, it is vulnerable to chemical modifications and strand breaks and lots of potential insults that can prove to be mutagenic. So in order to get around these problems, cells have a whole suite of enzymes referred to as RNase H enzymes, and they are each able, they occur in many classes, and they are able to fix different types of RNA-DNA hybrids. For example, some of the classes, like the RNase H2 class, can deal with a single nucleotide incorporated, a single ribonucleotide incorporated in a DNA strand. Some require a small chunk of the RNA to be present, and some of them don't even require a covalent junction and can still act on substrates without a covalent junction, whereas others cannot. And so it turns out that we know from some preliminary data that the lack of RNase H enzymes leads to increased mutation rates and genomic instability across all domains of life, from bacteria to humans. Defects in these enzymes are responsible for some genetic disorders in humans. They seem to play an important role in the evolution of bacteria because they, for example, shape the orientation of genes and the expression of antibiotic resistance genes. And so we would really like to understand the role of RNA-DNA hybrids in all of these processes. Before we can understand that, we have to measure where they are. Now, the standard in the field for measuring the locations of DNA-RNA hybrids has until very recently been an antibody called S9.6. This was an antibody raised against an RNA-DNA hybrid, and it does effectively pull them down, but it has several problems. It has a known sequence bias for AT-rich regions, which can bias the coverage that you get from pull-down experiments with this antibody. It has a strong affinity for double-stranded RNA as well as RNA-DNA hybrids, which can cause real problems for some experimental readouts, and it's expensive. So we came up with the alternative that instead of using this antibody, we would make and recombinantly purify an epitope-tagged RNA-DNA hybrid binding domain. So it's from an RNase H, but lacking the nuclease activity, and that we would be able to use this as a pull-down reagent to more efficiently and non-specifically find where there are RNA-DNA hybrids inside of cells. And so we compared a set of 3 methods to look at our ability to pull down RNA-DNA hybrids. We did a classic DNA-RNA IP using the S9.6 antibody. We did a refined version of the S9.6 protocol where we ethanol precipitated all of the nucleic acids before we went to do our pull down, or we used our hybrid binding domain in a pull down assay that looked like our refined S9.6 protocol. And what we did then was looked at our ability to pull down a R loop that was formed by a known R loop forming sequence called S-gamma3 when it was highly transcribed inside of E. coli cells from an inducible promoter. And so what we would want to see here is we're making 3 comparisons, either just background on the beads versus input in orange, the pull down versus input sample in green, or the pull down versus our beads without any affinity reagent, either the S9.6 or the HBD in blue. Ideally, what we want to see is the orange should be near 0, and the green and blue should both be high. And so this is the standard S9.6 protocol that a lot of people in the field still use. Doesn't seem to work very well. The refined S9.6 and the hybrid binding domain pull down, on the other hand, both work very well and roughly equivalently in terms of doing those pull downs. Now, another problem that I mentioned with S9.6 is it has this propensity to pull down double-stranded RNA as well. It only has about a 20-fold or less specificity for RNA-DNA hybrid versus double-stranded RNA. And so we wanted to look at how good our pull-down reagent is at pulling out only RNA-DNA hybrids. And so we made 2 sets of test duplexes, all as hairpins, where we have in each case a DNA duplex, an RNA-DNA hybrid, and an RNA duplex. But between set 1 and set 2, we flipped which of the sequences is the RNA-DNA hybrid and which is the double-stranded RNA, so we can make sure that there's no sequence bias creeping into our results. And so then we did a set of pull-down experiments, and this is what the inputs look like in both of these sets, where in this first set we have our RNA-DNA hybrid here, and then we have increasing amounts of double-stranded RNA down here. And on the other hand, in our second set, we have the double-stranded RNA up here. I apologize, the labels didn't turn out quite right on that for some reason. And the RNA-DNA hybrid down here, which is barely even visible in the input samples prior to pull-down. However, after the pull-downs, we see in both cases we recover the RNA-DNA hybrid wherever it was running, either up here or down here, regardless of its sequence, and we get virtually no double-stranded RNA present, even though this is present in 100-fold excess in the input samples. And so if we quantify the specificity for this, and I won't go into all the details, we end up with about 300-fold specificity or higher for the RNA-DNA hybrids over double-stranded RNA. So this is much better than what you're able to get with the S9.6 antibody. We also got concerned that maybe there would be an affinity for single-stranded DNA, which has been observed with some other pull-down reagents that are trying to pick out structures like this. So we did a separate set of hairpin experiments where we made DNA only with either a 5' or a 3' overhang, or we made, again, our RNA-DNA hybrid, and we again looked at the ability to distinguish the RNA-DNA hybrid in pull-downs from the DNAs that have overhangs. So here again is our input sample, and so what I've got shown here are the, my apologies, the DNA is larger, the RNA-DNA hybrid is smaller, and then when we get to the extracted samples, we are pulling out almost purely the RNA-DNA hybrid in favor of the much more abundant DNA hairpins that do have single-stranded overhangs. So this seems not to be a problem. So this pull-down reagent seems to be working very well for us at this point. And so we decided, okay, we've demonstrated this enough. It's time to go map where there are RNA-DNA hybrids inside of cells. And so what we're looking for in particular are things like these transcription bubbles that form potentially stable R loops. And if we look at the pull-down experiments, we can look separately at the mean pull-down signal on the sensed strand versus the anti-sensed strand. And this turns out to be really important because we get scatter plots that look like this one, where sometimes the sense and anti-sense signal are correlated with each other, and sometimes they're not. Before I talk about what that means biologically, I want to point out sort of the important secret weapon here, and that is when we do the pull-down experiments that we are doing here, we're looking at looking for structures that look like this, where we have a RNA-DNA hybrid, and we're also going to have some single-stranded DNA. And our pull-down reagent is going to specifically target this RNA-DNA hybrid after we have done some non-specific nuclease cleavage, which means this is literally the only thing that is ending up in our tube. And that absolutely requires us to have a single-stranded DNA sequencing prep that works well. And so one of the approaches that has worked for us is the Claret Seriously kit. That's the one we've used for a lot of this data. And so this gets to the what Claret is working on. I'm going to have some comments for things they could hopefully work on in the future because this has been very useful. We've tried some other prep kits. For example, there's a single-strand prep from Jay Shandur's lab that's a homebrew that works basically equivalently. At this point, though, we're using the Claret kit for the data that we're working on moving forward. And so going back now to the biological data, because we have single-stranded sequencing, we are able to look separately at the signal from the sense and the antisense directions. And that allows us to see whether we were getting enrichments of hybrids only on the template strand, only on the non-template strand, or both of them. And so if we look at the scatter plot of the sense versus antisense, we can see there's a sort of sea of genes where there is a pretty strong correlation, but it's hard to see it because there's just so much coverage here. There are some genes where the sense and the antisense signal are highly correlated, and I will get to those in a moment. And then there are some genes where there is a very strong bias, where the antisense signal is much higher than the sense signal, and that corresponds to what we would expect if we were enriching specifically for our loops as they are drawn here. And so sure enough, it tends to be a lot of strongly transcribed genes that are in this population that's showing the high antisense relative to sense. And so just to give an idea of what a couple of these look like, I'm showing here the occupancy plus 95% credible intervals from a really cool Bayesian analysis method, which I'll talk about at the end if I have time. So here is one very highly expressed region. And so we are seeing here that there is a bias where we're getting a lot more enrichment on the plus strand, which is the antisense strand, than on the minus strand. Here's another one. Again, we get higher enrichments on the plus strand than on the minus strand. You can see because this dotted gray line is at the same place in both axes, and almost all of the signal is above that line on the plus strand, and below that line on the minus strand. So what we see is where there is a lot of transcription happening, the R loops are all forming due to transcription. On the other hand, we can also look at genes that have very strong levels of hybrid formation, but it is roughly the same on both the plus and the minus strand. And it just so happens that a lot of these are in these extended protein occupancy domains that I was talking about before. So for example, the WA operon is exactly the one that I was showing some protein occupancy landscapes for earlier in my talk with the HNS and the STPA. And so here is what the zoomed in enrichment plots look like in these regions. And in this case, we see roughly similar levels of RNA-DNA hybrid signal on the two strands. And so this is going back to, for the WA genes, for example, here are the WA genes. Just a reminder, this is what our protein occupancy landscape looked like in wild type cells in the WA operon. And so what we think is going on here biologically is the presence of this heterochromatinized region does not deny access to RNA polymerase, but it prevents the polymerase from actually being able to elongate. And so what you get instead is you get attempts at transcriptional initiation followed by abortive transcription. And the RNA polymerase remains trapped, forming these hybrids, but not able to elongate until you open up that HNS filament and then allow transcription to occur. So this is a mechanism for silencing regions, but allowing poised polymerase to be present that can, under some conditions, allow transcription to occur. So another interesting set of findings that we have relates to what happens when we delete the RNA chaperone HFQ. So HFQ is known to act on small RNAs in E. coli and help them find their targets. And what's very interesting then is if we look in the vicinity of known HFQ clients in a delta HFQ strain in green versus wild type cells in blue, we see strand-specific hybrid accumulations on the template strand in this region when we, consistent with our loop formation, when we've deleted HFQ. So it seems to be disrupting transcription in these areas and causing polymerase stalling, which is giving us these strand-specific pileups of RNA-DNA hybrids. Again, the ability to get strand-specific DNA sequencing data is absolutely essential here for us to figure out what is going on. Here's just another example. This is the case of CYAR, where we also see some increases in RNA-DNA hybrids in a delta HFQ strain. Here's another known target. This is the OMP-X small RNA. So this seems to be a pretty common trend. So what we've gotten from our applications of the hybrid binding domain-based sequencing in E. coli is hybrid pull-downs allow us to enrich the hybrids genome-wide. We need to have strand-specific sequencing in terms of figuring out where they are. And we see two different biological mechanisms that appear to be giving rise to the hybrids. At very strongly transcribed regions, we see things consistent with R loops from elongating transcription, where there is a strand-specific formation of hybrids being caused by the RNA polymerase forming a transcription bubble. On the other hand, at silenced regions, we see bidirectional accumulation of hybrids that looks like it's due to abortive transcription and early transcriptional termination occurring. So I think I need to be done in about 5 minutes. And so I'm going to skip a last little part of the biology because I wanted to also talk briefly about some software we've recently developed that is highly complementary to the sequencing approaches that I'm talking about here. And so just skipping forward briefly, because I do have more data. So the first tool that I'd like to make people aware of, because I think it could be very useful paired with single-strand sequencing, is something called Enricherator. And so Enricherator is a computational post-processing tool that we've developed that does Bayesian analysis of chip-like pull-down data and allows you to get a couple of benefits over any kind of classic method for analyzing chip-like data. So first of all, there are credible intervals, which are kind of like confidence intervals, but in Bayesian statistics, built into the results. So if I go back a slide, we can see there is some fuzz around each of these signals. This is actually a 95% credible interval. So you're getting your significance testing happening as a part of your primary analysis. In addition, there is a shrinkage prior applied to this analysis, which means you don't have to do any corrections for multiple hypothesis testing. This is already baked in. And so this is an example of what it looks like with one of our credible intervals. In blue is an Enricherator analysis. In red is a more classic analysis of chip data that's just based on doing log ratios with a pseudo count. And we can see that the naive analysis both gives you some weird discontinuities in the data, and it also doesn't give you the kind of credible intervals that we're able to get with Enricherator. The other strong benefit, I think, comes from the fact that Enricherator allows sharing of information across neighboring genomic loci, which helps to properly account for the presence of finite-sized fragments in DNA sequencing samples that are going to give rise to some of the signal. And then two other technical things that I think are useful. Enricherator natively handles stranded DNA sequencing data, so it can do these occupancy profiles on data coming directly out of any strand-specific DNA preparation method, which is useful to the quantitation that we're doing. It's also compatible with having spike-ins, which allows you to do quantitative ChIP-seq experiments. And one other method I'd like to point out is one of the things that we've noticed over the course of looking at these highly abundant nucleoid-associated proteins in bacteria, but it's a problem in all organisms ranging up to humans. We often try to figure out the binding preferences of DNA binding proteins based on sequence motifs. You get a sequence logo or something like that. But sometimes it's not actually sequence, but shape that is recognized by DNA binding proteins. DNA, after all, is really a three-dimensional object, not just a set of letters. And so it has parameters such as the minor groove width and the twist of the helix and things like that, that are dependent upon DNA sequence, but don't always map, dependent upon the sequence, but don't always map directly to the sequence. And what I'm showing here is if you take all possible 5-mer DNA sequences, the distances between their sequences are shown in the lower triangle. Their distances in the space of shape features are shown in the upper triangle. And there's a very different picture in terms of what sequences are similar to each other if we look through the lens of shape, which is often what proteins are seen when they try to bind DNA. So we've developed a method called ShapeMe, which is a computational method that can take data from pull-downs, be they chip or RNA-DNA hybrid pull-downs, or RNA-seq data where we look at regulatory regions, and we're able to identify shape motifs in the DNA that are closely associated with binding profiles. And so to give an idea of how this behaves, we did an experiment where we took a bunch of different human transcription factors for which high throughput data on their binding preferences is available, and we tried generating either shape motifs, sequence motifs, or joint motifs for those DNA binding proteins. And so you want to be on this graph, farther to the right is better. The green is the shape motif performance. The red is the sequence motif performance. The blue is both of those motifs to get together. So if we look at a factor like the top one here, POO5F1, we can see that the performance is roughly the same for the shape motif and the combined motif. And that means this factor really only cares about shape. And the sequence motif does substantially worse. Here's another one, and here's another one, cases where it is really all about binding shape. There are other factors that really do seem to care about sequence. Here's an example. So the JUND transcription factor, we see that the performance of the shape motif is worse than either the both or the sequence motif. And so if we look across different DNA binding proteins, we can categorize them where some really seem to care only about shape, some care about sequence, some care about both. But what ShapeMe is able to give us then is this type of shape motif, and this is what they look like. And I think for the sake of time, I won't go into any more detail on what this looks like, but this is what these shape motifs look like. They are often higher performance than corresponding sequence motifs in terms of identifying where a DNA binding protein is going to bind. And we think they'll also be useful, although we're going to be working on this, for designing DNA binding sites and also understanding things like off-target cleavage of CRISPR systems and other things that are very likely DNA structure dependent. So the last point I would like to make, and then I will close, is I had a couple of notes on some future applications for related things that we're interested in, and hopefully I can convince the Claret folks to work on in some cases. So we're interested in using single-stranded DNA sequencing to let us do higher resolution profiling of protein DNA interactions because we can see the strand-specific cleavage sites. It lets us map binding sites more tightly. We'd be really interested in finding ways to simultaneously sequence associated RNA and DNA. So for example, and I've talked to Varsha about this before, if you take one of these RNA-DNA hybrids that we're able to pull down, could we simultaneously sequence the RNA and DNA pieces of this and know that they came from each other? And more generally, can we come up with good single-stranded prep methods that let us identify junctions between nearby nucleic acids, either RNA-RNA interactions or DNA-DNA contacts as are found in high-C maps? How well can we incorporate modified base detection into these single-strand workflows? And when can we get some kits for other sequencing platforms? So those would be, I guess, my questions to the hosts. At that point, I'd also just like to thank my entire lab and all of our collaborators who worked with us on the projects I was showing you today. And I'd be very happy to take any questions that you all have.
Thank you, Dr. Freddolino. That was a very insightful presentation. I believe we have one question. If you can put any of your additional questions in the Q&A chat, that would be great. And I have Michaela Ford. She has a question. I'm not sure if I can allow. Michaela, would you be able to add your question into the Q&A comments? I'm not sure if I can unmute you.
So I do see one anonymous question in the Q&A.
Oh, yeah.
In the Q&A, do we see more DNA-RNA hybrid in regions between divergent promoters? And yeah, there's a lot of pileup there. And in general, the hotspots tend to be promoters, especially highly expressed promoters, and also 5' UTRs tend to have pileups as well, and especially the very strongly transcribed regions like ribosomal RNA operons. But yes, there's a big pileup around active divergent promoters.
Andrew Measham. Oh,
yeah, so should I read it? I'm sorry, go ahead.
Oh, I can read it. Could you revisit the comparison of performance between the common antibody-based method of isolating DNA-RNA hybrids and their novel method using the inactive RNase H? What indicates better performance with the latter method?
Yeah, so I would say as far as the performance being better, so here's the key performance comparison where we have direct head-to-head comparisons. This would be the S9.6 antibody done using pretty standard protocols. This is a highly refined S9.6 protocol. This is our hybrid binding domain method. So remember, we want the green and blue to be high. We want the orange to be low. So I would say this seems roughly equivalent in our hands in terms of a really well done S9.6 pull down versus the HBD. I believe the superiority of the HBD comes from its much higher specificity. We're talking about at least 300-fold and possibly 1,000-fold for hybrids over double-stranded RNA, whereas you're in the ballpark of 20-fold with the S9.6. And in addition, we see no signs of the kind of sequence specificity in the hairpin experiments that I was showing later for the hybrid binding domain, whereas we do know from the literature that that exists for the S9.6. So I would say if you do S9.6 well, it's roughly equivalent in terms of pull-down efficacy, but not as good in terms of doing things like distinguishing between RNA-DNA hybrids and double-stranded RNA. I hope that addresses your question, but please feel free to follow up if you'd like any more information on that. I see Varsha has a hand up. I don't know if
yes,
I should call on people or what.
Yeah, I have a couple of questions. One is to do with the ShapeMe software. I was curious to find out if the position of those various motifs along the strand, does that have an effect on its function, its interaction, and so on?
Yeah, so that's a really, sorry, go ahead.
Especially if it is terminal or central in position.
Yeah, that's a really great question, and that is used in some sequence-based motif finders. At present, we do not account for the location within the window of the motifs, but we account for both orientation and the number of the motifs. And both of those do seem to matter for some factors, especially factors that we already know from the literature require multiple binding sites. That does indeed turn out to be important. We have talked about incorporating the actual positioning of the motif in a window in a future version, but right now we're just restricted to encoding the count and the orientation. And we do that feeding into all of our motif finding.
The other question was, given that you are getting all these insights into this mechanism of DNA-RNA hybrid, is there any idea about using this as a druggable mechanism, to use it in, whether it is to revert antibiotic resistance, anything from that perspective?
Yeah, it's a great question. So the RNase H enzymes, unfortunately, the classes of them are fairly conserved across domains of life, so they're not going to be a great drug target themselves because there would be too high a possibility of, or I think it would be very hard to discriminate between host and bacterial copies of these enzymes. I didn't get time to talk about this, but the presence of RNA-DNA hybrids seems to play a major role in shaping the genome because it actually dictates the orientation of a lot of the genes in the genome, where if you have a head-on interaction between the replication fork and a highly transcribed gene, it tends to cause damage. That's been shown by other labs, most notably Horomeric, and this is an R-loop dependent mechanism. So a lot of highly expressed native genes will be co-oriented with replication. For example, all of the ribosomal RNAs in bacteria are. Now, the reason that is potentially important is when horizontal gene transfer occurs, which is a common way for antibiotic resistance to spread, the orientation of the genes is initially random. And so if there were ways to partially disrupt the repair of these RNA-DNA hybrid-induced lesions that can occur during head-on conflicts, it might potentially inhibit horizontal gene transfer, but that's a bit of a stretch, and I don't have a great mechanism for how you could do that, again, without also hitting human enzymes that play important biological roles.
Great. Thank you so much for answering all those questions. Do we have any additional questions that anybody would like to put in the Q&A? If not, if there's no more questions, please join me in thanking Dr. Lydia Freddolino today. And you can write to her with any additional questions, and we will be sharing this webinar with all the attendees, along with her contact information, as well as Claret's contact information.
Thank you all very much for your attention. I appreciate your time.
Thank you.